Hey there, in this video I'm going to talk you through a word example for sex linkage. The specification for the AQA spec, you need to use a fully labeled genetic diagram to interpret and predict the results of crosses involving sex linkage, autosomal, multiple and epistasis. So we're going to focus on sex linkage. Any gene that appears on the sex chromosomes are referred to as sex-linked, as opposed to the other 22 chromosomes called autosomes, and those genes are referred to autosomal linkage if they are linked together. We'll talk about that later. Now, in this diagram, which you probably can't see too well in the video, but if you Google, you'll find something fairly straightforward. Here we have the X chromosome and the Y chromosome. And the X chromosome is longer and has different genes in the same loci as the Y chromosome. So if a man receives an X chromosome from his mother that has, well, there's hemophilia right there, and it is the hemophiliac gene, he has no homologous gene to counteract its expression, and will end up being a hemophiliac male. So let's take a look at that. Okay, so let's do a cross for a carrier female and a normal male. So parental phenotype, carrier female, crossed with a normal male. Parental genotype. So since this is sex-linked, we need to use our X chromosomes and our Y chromosomes to represent the sex of the cross. And then we've got carrier female, so she has a normal gene for the blood clotting protein and an allele, or sorry, norm, that was allele, and a disease allele here, so she's a carrier or heterozygous. The male is normal and his Y chromosome doesn't have that gene loci. So gamete genotypes, here we go, and there is the disease allele. Okay, so let's put mother on top of our Punnett square. We'll put father down the side. Mother X superscript H, X superscript little h, X superscript big H for the father, and Y. So I'm going to do the sex determination first. X and X, X and Y, which gives us our 50% chance of male or female offspring. And now I'm going to bring in my alleles. Just use a different color here so it stand out a bit better. There's big H, superscript big H, little h, and we've got big H there and a little h there. Okay, so what are our genotypes? We have that one, so two girls, and boy and a boy. Phenotypes, normal female, homozygous dominant for the normal condition. And we have a carrier female, she's heterozygous, doesn't express the disorder. And we have a normal male and hemophiliac male. Ratio of phenotypes. So we've got normal female 2, normal male 1, hemophiliac male 1. If we can look solely at the inheritance of the alleles and the disease hemophilia, then that becomes normal, so that would be 3 to 1, not taking account of the sex determination. So basically a normal monohybrid cross, and that's how it's done.